Hello friends, I'm Dr. DP Bora. Welcome to my YouTube channel, The Microbiology Digest. So today we'll be discussing a new topic, another topic of microbiology, that is plasmids and antibiotic resistance. So already we have discussed in the bacterial structure that plasmids, they are present in most of the bacteria. Today we will be discussing in detail about the plasmids and what is their significance in the term antibiotic resistance. So now what is bacterial plasmids? So these plasmids, they are small circular pieces of extra chromosomal DNA that is that exist in bacteria, many species of bacteria, and they can replicate independently of the host chromosomes. Means plasmids are small circular pieces of extra chromosomal DNA which they can replicate independently. Okay, so the term plasmid, it was first used by one scientist, Joshua Lederberg, in the year 1952. Now the plasmids, they are usually, it may be their size may range from 1 to 1000 base pairs, and many bacteria, as I said, they carries this plasmids. So in the diagram, you can see plasmids diagrammatically, it has been illustrated. This is an extra chromosomal component. This is a circular DNA, piece of DNA, and it is present in the bacteria, which is independent, which can replicate independently of the bacterial DNA. Now, what are the properties of plasmid? As we have already discussed, they are small double-stranded DNA molecules, and they are usually circular. They are capable of independent replication, we have already discussed. And plasmid have relatively few genes, generally less than 30 genes. The number of genes that may be present in case of plasmid is generally less than 30. They occur naturally in bacteria, and they carry genes that are useful, but they are not essential for the survivability of the bacteria. Is it clear? So they carry many genes. They are useful, but they are not essentially required for survivability of the bacterium. Some plasmids, they even contain some genes that can help in the transfer tube. Already we have discussed during the conjugation process. So they helps in conjugation process also. So plasmids, they can be eliminated from the host bacteria. There is a specific process. This process is called as curing. Curing, curing is the process by which plasmids can be eliminated from the host bacterium. Now, the classification. There are many ways by which you can classify plasmids. First is based on the ability to transfer to other bacteria. So based on this party, two types of plasmids exist. First one is the conjugative plasmid. So these plasmids, conjugative plasmids, they contain a specific gene. This gene is called as trazine, or they have the capacity to transfer plasmids to other bacteria. Because of this trazine or transfer gene, so they have the ability to transfer plasmids from one bacterium to another bacterium. Then second type is the non-conjugative plasmids. So they are not capable, as they do not contain trazines. They are not capable of initiating a conjugation. Okay, so they can be transferred, but always there is requirement of the conjugative plasmids. So example of this plasmid is the resistance plasmid, or R plasmid we say. And example of conjugative plasmid is the F plasmid or fertility plasmid. Okay. Now, based on function, there are 5 different types of plasmids can be or maybe there. First type is the R plasmid or resistance plasmid. So, what are R plasmids? They have the genes for resistance against antibiotics. So, they are historically they are called as R factors. Okay, and these type of plasmids they are mostly responsible for development of resistance against antibiotics. They are plasmids. Second type of plasmid is col plasmid. So they are responsible for production of colicins. There are some specific compounds against they are antibacterial compounds produced by some species of bacteria. So these plasmids they are responsible for this function or production of colicins. They are called as colplasmids. So third type of plasmid is the fertility plasmid or F plasmid. So they are responsible for conjugation process in bacteria. Fourth plasmid is the virulence plasmid. So they contains this type of plasmids. They contains a specific gene. The name of the gene is Vir gene, VIR. Okay. So because of this presence of this plasmid, virulence plasmid, these plasmids can convert a bacterium to a pathogen, or this confers the ability to a bacterium to cause a disease. Then fifth type is the degradative plasmid. Degradative plasmid, they are able to degrade unnecessary substances like toluene or salicylic acid. So this is toll plasmid, for example. In case of Pseudomonas organisms, they contain a toll plasmid. So these plasmids, they are capable of degrading unnecessary substances like salicylic acid, toluene. So these type of plasmids are called as degradative plasmid. Now, use of plasmids, as I said, plasmids are one of the very important tool, mostly in today's era of molecular biology. So, few uh, important use of plasmids are listed here. Plasmids are used in genetic engineering. As I said, they can be used to amplify or produce many copies of a particular gene. So, they are used in molecular cloning as a vector. So, plasmids are a very good vector. Then they can be used for production of proteins. Then they can be used in gene therapy, and plasmids are also used to study the disease models. So these are some of the important uses of plasmids. Now next part of today's discussion is the antibiotic resistance. So before that, let us understand what is antibiotic. So antibiotics are some chemical compounds produced by microorganisms that can kill or inhibit the growth of some another microorganisms. Means these are produced by microorganisms and they can kill or inhibit the growth of other microorganisms. So antibiotics, after discovery, it has almost revolutionized the entire uh, medicine. And the first antibiotic which was discovered by Alexander Fleming in the year 1929, it was penicillin. So what are the properties? They can kill or inhibit the growth of pathogens. They cause no damage to the host. Generally they cause no side effects to the host. They are stable when stored in solid or liquid form. They remain in specific tissues for longer duration to be effective, and they kill pathogens before they get muted or they develop resistance to it. So these are some of the properties of antibiotics. Now, how they work? So there are specific methods by which antibiotics works. So first is the inhibit the cell wall synthesis in bacteria. Then they may inhibit the protein synthesis. They may inhibit the nucleic acid synthesis. They can cause injury to the plasma membrane. or they may inhibit the synthesis of essential metabolites. These are some of the pathways through which these antibiotics they works to kill or inhibit the growth of bacteria. Now, resistance to antibiotic in today's world, this is one of the most widely discussed topic, that is the antibiotic resistance. So, antimicrobial resistance, it is the ability of a microbe to resist the effects of medication. So, this is a general term. So, when a microbe it attains the capacity or ability to resist the effects of medication that was once used to treat these type of infections, so that stage is called as antimicrobial resistance. So antibiotic resistance, it is a part of antimicrobial resistance, and antibiotic resistance, it is used when we apply antimicrobial resistance to bacteria and when we take into consideration of the antibiotics, then we call it as antibiotic resistance. So it is one of the biggest threats to the global health because of uh, rising concerns about the development of antibiotic resistance by many organisms. It also relates to the food security and the entire development of the society. So antibiotic resistance can affect anyone, any age or in any country. So antibiotic resistance occurs naturally, but the misuse of antibiotics in humans and animals is one of the major factor which is accelerating the entire process. So there are certain issues. Antibiotic resistance leads to longer treatments and it incurs higher costs, and also it can lead to increased mortality in case of the affected host. Now, what are the mechanisms of antibiotic resistance? So, and when a particular organism develops resistance to any drug or any antibiotic, so what may be the possible ways? So, first way is the organism will prevent the antibiotic from reaching its target. So, as we have already discussed, antibiotics will work through certain specific pathways, or they will have a specific target in the bacterial cell. So they may affect the cell wall, they may affect the plasma membrane. So organisms, they will develop some system which will prevent the antibiotic to reach its target. So this is one of the mechanism of development of resistance. Then second option is the organism will prevent the antibiotic to bind to its target. So to exert is and its effect, so antibiotics must bind to its target. Okay, so what will happen if if the organism has developed resistance, then it will prevent the binding of the antibiotic and the target. Then there may be inactivation of the antibiotic or the drug before reaching its target, and there may be genetic transfer of the antibiotic from or antibiotic resistance from one bacterium to another bacterium. So these are some possible ways by which an organism may develop resistance against antibiotics. Now some examples of antibiotic resistant organisms. So vancomycin resistant enterococci, this is in short, it is written as VRE. Then MRSA, it is methicillin resistant Staphylococcus aureus. Then penicillin resistant Streptococcus pneumoniae. Then MDR-TB. These are some of the uh, important organisms which have already developed resistance against some of the antibiotics, and they are causing major concern to the society. Now, how we can prevent this development of antibiotic resistance? So first, we should keep in mind that antibiotics should be the last line of defense. Means if other procedures have failed, then only we should go for use of antibiotics. Then we should take antibiotic only when it is prescribed by a physician or a doctor. Then if antibiotic is prescribed, we should ensure that the course of the antibiotic is completed. So if these things are followed, so there is a possibility that there may be, we can prevent the development of resistance by the antibiotics. So lastly, there's a much discussion needed to prevent this global problem of development of antimicrobial resistance or antibiotic resistance. For this, a proper action plan is needed, not only in the regional level, national level, international level. There should be a proper action plan, educational campaign in various levels, then strengthening the legislation. There should be a strong legislation uh, for the suppose use of antibiotics. Then all different line departments, government departments, and other NGOs, they should come into the uh, front for to develop some awareness among the public, so that this concern rising concern about the antibiotic resistance can reach to one and all in the society, so that people will know about this global problem. So this is what I wanted to discuss today. So with this, I hope this information will be helpful, and if you find these are helpful, please subscribe to my channel, and do share with your friends, so that it the informations included here will reach you at the earliest. So with this, thank you very much. Thank you. Bye bye.